Hello students, welcome to today's class. In today's class, in photosynthesis in higher plants, we are going to study about two major topics. One is your Kelvin cycle and another one is your Hatch and Slack cycle. So a Kelvin cycle at first we will be taking on. Now whether it is a C3 plant or a C4 plant, Kelvin cycle occurs in all of them. Now what do you mean by a C3 plant and a C4 plant is that those plants in which while carbon dioxide fixation the first stable carbon compound is a 3 carbon compound, we call that type of plants to be C3 plants and the cycle to be the C3 cycle or the Kelvin cycle, known after the name of the person, that is Melvin Kelvin, who has found this pathway. And of course those plants in which the first stable carbon compound in the carbon dioxide fixation is a 4 carbon compound, which is oxaloacetic acid, we call that cycle to be the C4 cycle, or that is your sometimes Hatch and Slack pathway, depending upon the name of the person who has found it. Okay. Now at first, the primary carbon dioxide acceptor which we have to learn today is in case of your C3 plants or in case of your Kelvin cycle, the primary acceptor of carbon dioxide is a 5 carbon compound. The name of that 5 carbon is RUBP, ribulose 1,5 bisphosphate. Okay, so ribulose 1,5 bisphosphate is a 5 carbon compound which accepts from the atmosphere carbon dioxide, and the first step of the C3 cycle takes place, which is called carboxylation. So the fixation of the carbon dioxide takes place with the help of a 5 carbon compound RUBP, resulting into the first stable carbon compound in case of these kinds of plants, which is the C3 plants, and normally the mesophytic plants which is growing around, that is your 3-PGA, 3-phosphoglyceric acid. This is the first stable carbon compound formed in case of C3 plants. Okay. And as it is a 3 carbon compound, so that is why it is called a C3 cycle. Now see here, it was 5 carbon compound and 1 carbon dioxide is accepted by it, so it becomes 6. So ultimately it will be 2 molecules of 3-PGA because it's a 3 carbon compound, so we have 6 carbons. So 6 carbon means 2 molecules of this 3 carbon compound, that is your 3-PGA, is formed as a stable compound, and this is known as your carboxylation, which is the first step of your that is your Kelvin cycle. Next, after this is we have come to know from our the light reaction part of the photosynthesis that ATP and NADPH is generated. So that ATP and NADPH will now be utilized by this 3-PGA for a reduction and phosphorylation to result into formation of triose phosphate, which will form a stable compound or the glucose or sucrose, which undergoes polymerization to form the starch. And the rest will be taking part in the regeneration of RUBP, because this cycle, if it is to be continued, because if 1 carbon is taken up, and then it will be the photosynthesis step, the dark reaction step will not follow further, isn't it? But it's a continuous process. Light reaction and dark reaction are continuous process. So until and unless this RUBP is regenerated back, who is going to take the next carbon dioxide from the atmosphere? So the regeneration of RUBP is must. Some of them takes part in the formation of sucrose and the rest others takes part in the regeneration of RUBP. Now this RUBP, which has accepted the carbon dioxide from the atmosphere, it takes place in presence of an enzyme. The name of that enzyme is called as your Rubisco. Okay, this is called as your Rubisco, RUBISCO. Now Rubisco. Now where is this found? In case of C3 plants, it is found in the mesophyll cells. The mesophyll cells are having this Rubisco. So as a result, since Rubisco is there, so the RUBP in presence of this enzyme Rubisco is capable of fixing the carbon dioxide. But about the C4 plants, which we are going to see next, they do not have this Rubisco in their mesophyll cells. So where are they present? They are present in the bundle sheath cells of the C4 plants. Now what are these C4 plants? These can tolerate a high temperature These are the temperate plants. These are plants like that of your sorghum, plants like that of your maize, plants like that of your sugarcane. So these type of plants, if you just make a section of the leaves of these plants, you will find that the mesophyll cells are somewhat bigger and they are thick walled, and they are just forming a wreath. Wreath means a kind of, you know, circular structure, and the bundle sheath cells are protected inside. So since these cells do not have the rubisco in them, but they have another enzyme, we will be coming to it, and then gradually when you know, temporary acid is formed, which is a C, of course that is your C4 carbon, that is OAA, oxaloacetic acid, then gradually that will be, you know, decarboxylation will take place in the bundle sheath cells, because in the bundle sheath cells they have the rubisco. So they have actually two enzymes, one for, you know, fixing the carbon dioxide in the mesophyll cells, but it is of no use because there the formation of glucose or the sucrose cannot take place, because there is Kelvin cycle cannot go through. So it has to come finally to the bundle sheath cells through those plasmodesmata, and transportation will take place, and then they will come to the bundle sheath cells, and finally in the bundle sheath cells they have the rubisco, which will be helping them to fix the carbon dioxide by the process of that pathway of Kelvin cycle, and C3 acid will be regenerated, which again will be transported back to the mesophyll cells for the formation of the carbon dioxide acceptor. Now here it was a 5 carbon compound, here it will be PEP, that is phosphoenol pyruvate, which is produced in the, that is your mesophyll cells, and that is the enzyme which is associated for the PEP, that is phosphoenol pyruvate, to fix the atmospheric carbon dioxide, is called your pepcase. Okay, so that is the difference between that of your C3 and the C4 pathway, which we will be again discussing, of course. But let us come back to this C3 cycle or the Kelvin cycle. So triose phosphate, from this some of them take part in the formation of sucrose, and finally by the help of polymerization they form the starch, and rest others they again in of course by the utilization of ATP, which was formed in your light reaction, results into formation of that 5 carbon compound, which is your ribulose 1,5-bisphosphate. Fine. Now see one thing. Now here one carbon is taken up by this RUBP, but we know that glucose is C6, isn't it? C6H12. So this cycle has to turn 6 times for the formation of a glucose molecule. So 6 carbon, for the fixation of the 6 carbon, this has to undergo 6 turns, and for 1 carbon fixation, that is the reduction step which is taking place, NADPH is reduced there. So in this reduction step, this 3-PGA, okay, this 3-PGA, for 1 molecule of carbon dioxide, it requires for phosphorylation 2 ATP molecules and 2 NADPH molecules. So if it is 1 carbon, so I said that it will undergo 6 turns. So for 6 carbon, definitely it will be how much? So for 1 it is 2, so 6 2s are 12, so it requires 12 ATP. It requires 12 ATP for the 6 turns of this cycle, so as to form the glucose molecule. Likewise, for reduction purpose also it requires 2 NADPH. So since for 1 carbon it requires 2 NADPH, so for 6 carbon dioxide to be fixed, it requires how much? 6 into 2, 12 NADPH, isn't it? 12 ATP and 12 NADPH. Fine. Here it is forming the glucose or the sucrose and the starch. But again, for the regeneration of RUBP, 6 times this cycle is undergoing. So here ATP is utilized for 1 turn. For 6 turns, 6 ATP will be utilized. So here it was 12 ATP, because for 1 carbon dioxide fixation it required 2 ATP molecules. So 6 turn means 6 into 2, that is 12, and here it is going 6 times. So here 6 ATP will be utilized. So here 12 and here 6. So it is becoming 18. So 18 ATP will be utilized. Okay, 18 ATP will be utilized for the formation of 1 molecule of glucose. So 6 carbon dioxide, this is, you can say, the photosynthesis chart in and out. You will see it is given in your book. So 6 CO2 molecules will be required for the formation of 1 molecule of glucose, okay, and 18 ATP is required for the formation of 1 molecule of glucose. How? Because for 1 molecule of carbon dioxide fixation it requires 2, so 6 turns means 6 into 2, so that is 12, and here it is 6 times going, here it is 1, so 6 times it is going, so it will be how much? Here it will be 6, so 12 and 6, it will be 18. But NADPH, if you consider NADPH for one fix, that is your reduction of carbon dioxide molecule, it requires 2 NADPH. So for 6 carbon dioxide, it will require 6 into 2, 12 NADPH. So in and out chart is explained in that way. In 18 ATP and out will be 18 ADP, adenosine diphosphate, okay. Similarly, in will be your 6 CO2, out will be 1 molecule of glucose, and similarly for NADPH it will be your 12 NADPH will be utilized for the formation of glucose, and finally it will give out will be your 12 NADP plus, okay. So this is the total thing about your Kelvin cycle, and why it is called C3 cycle. Sometimes in exam this question comes because the first stable carbon compound is a 3 carbon compound. Where does the Kelvin cycle takes place in case of plants? Kelvin cycle takes place in the mesophyll cells. Why? Because in the mesophyll cells of the C3 plants, they have the enzyme which is called as your rubisco. Okay. Now let us come to the Hatch and Slack pathway, or the C4 cycle as it is popularly known as. Now from the atmosphere, the carbon dioxide enters. Now this carbon dioxide is accepted by another carbon dioxide acceptor which is present in the mesophyll cells, which is called as your PEP, PEP, phosphoenol pyruvate, okay. Now this phosphoenol pyruvate fixes the carbon dioxide entering from the atmosphere and makes it a unstable carbon compound, which is a 4 carbon compound. Now this can be aspartic acid, this can be malic acid, and then this 4 carbon compound is transported to the adjacent cell. This is a bundle sheath cells, and they are so compactly arranged. There is no gap also in between the mesophyll cells and the bundle sheath cells in case of the C4 plants, like I told you, the sugarcane plants or the sorghum plants and all. So it is transported through this that is your plasmodesmata, or the that is interconnection in between the adjacent 2 cells, and then it is converted to that of your oxaloacetic acid, which is a 4 carbon compound. Now this oxaloacetic acid will undergo the process of decarboxylation. D means emission of 1 molecule of carbon dioxide. So decarboxylation takes place. So when decarboxylation takes place, carbon dioxide is released. Now this carbon dioxide which will be released, it will undergo the normal C3 cycle or the Kelvin cycle. Why? Because here we have the rubisco, or the RUBP is also present there, which will be now accepting this carbon dioxide, and then 6 turns will go on, and finally from this oxaloacetic acid, if 1 carbon dioxide is removed, because it was a 4 carbon compound, so 3 carbon compound or 3 carbon compound acid will be released. Now this will be again transported back to the mesophyll cells. Now in the mesophyll cells, again regeneration of PEP will take place. Why? Because if the regeneration of PEP does not take place, then this cyclic process of the C4 cycle cannot take place, as it is the same case in case of RUBP. If RUBP is not regenerated in case of C3 plants, the next carbon dioxide molecule from the atmosphere cannot be fixed. Okay. So this is about your normal difference between that of the Kelvin cycle and the Hatch and Slack pathway. Here everything is taking place in the mesophyll cells, okay, but here we need 2 different cells, as because these plants have a definite arrangement of cells. We call them as Kranz anatomy. That is, the bundle sheath cells are surrounded by that of the mesophyll cells in a wreath like manner, in a circular necklace like manner around them. So that's why they have to follow this kind of pathway. And the plants which are the C4 plants, they are much more economical in terms of production of your food or glucose, and they do not follow the photorespiration path, that is process, unlike the C3 plants. So C3 plants can undergo photorespiration, but C4 plants do not undergo the process of photorespiration. We will come to the photorespiration, which is sometimes also known as your C2 cycle. because the first carbon compound formed there is a 2-carbon compound. Okay, so here it is 3-carbon compound, so C3 cycle. Here it's a 4-carbon compound, so it's a C4 cycle. So that one, photorespiration, is sometimes known as your C2 cycle. The reason is because the phosphoglycolate which is formed, it is a 2-carbon compound. Okay, so and that photorespiration never takes place in case of plants which are the C4 plants, which have the Kranz anatomy. It takes place only in case of the normal plants which have the normal mesophyll cells, not having the bundle sheath cells inside, and they have the rubisco, normal rubisco. So they will undergo the process of photorespiration, but these type of plants will never undergo the process of photorespiration. So that's all for today's class of photosynthesis in higher plants, where we have studied about the Kelvin cycle and the Hatch and Slack pathway. Next we have just photorespiration and that is your the factors affecting photosynthesis. That's all for today's class.